Okay, thanks very much for a great talk, Robin. I want to start by saying thank you to David Hood and the organizers of the conference for inviting me to give this presentation. The title of my talk is SR Calcium Pump Modifications in Human Neuromuscular Fatigue. So this is a brief overview of what I'll be talking about today. I'm going to start by talking about models of SR calcium pump dysfunction to demonstrate how slowing of SR calcium uptake specifically impairs skeletal muscle performance and can cause disease. And then I'll cover some studies that have assessed SR calcium pump modifications with fatiguing exercise in humans. And I'll finish by talking about a novel intervention to prevent exercise-induced SR calcium pump dysfunction in order to assess the role of calcium pump modifications in human neuromuscular fatigue. So to start, the definition of muscle fatigue simply is a reversible decline of muscle performance during activity, and this includes decreases in force, speed, and power generating capacity, as well as decreases in the rates of force development and force relaxation. Most recovery occurs within the first hour, but there's also a slowly reversible component that can take several days to recover, so-called prolonged low-frequency force depression. So how could SR calcium pump dysfunction and slowing of SR calcium reuptake impair skeletal muscle performance or cause fatigue? Well, first, theoretically, it could slow the rate of force relaxation. And this is really nicely demonstrated by Nicholas Place and colleagues, showing how slowing the rate of force relaxation can impair performance of intense running exercise, even if force generating capacity is fairly well preserved. So in this example, you can see decreases in the rate of force relaxation leading to decreased stride frequency, and assuming that the stride length remains constant, this would reduce running speed by 3/5 of the original. The second possibility is that circuit dysfunction may contribute to the increased intracellular calcium that occurs during fatigue. As Robin mentioned, this is also due to RYR fragmentation and increases in RYR leak. And we know that increases in intracellular calcium can cause calcium-induced proteolysis of junctophyllin, which is shown here in red, causing excitation, contraction, uncoupling, and decreased calcium release, as Robin and colleagues reported in their 2013 JPhysiol paper. Now, whether circuit dysfunction plays a role in this process is really not well established. It's theoretical at this point. But what is well established, as I'll show you next, is that genetic models show that specific defects in SR calcium pump function cause impaired skeletal muscle performance and can cause disease. And one of the best examples to illustrate this is a genetic disease called Brody's disease, which is a rare inherited disorder of skeletal muscle caused by SERCA1 deficiency. And this results in exercise-induced impairment of skeletal muscle relaxation, stiffness, and cramps. And as you can see in the photos of this Brody's patient, who is forcefully shutting his eyes, clenching his fist on the right here, and then is asked to relax and open his eyes and open his hand, he's unable to do this very quickly. So this is very slow going from right to left. And in fact, these patients would struggle to climb a single flight of stairs without completely cramping before reaching the top. And when I was a postdoctoral fellow in David McLennan's lab at the University of Toronto, we generated the SERCA1 knockout mouse as an animal model to study Brody's disease. And it turns out that knocking SERCA1 out of mice impairs diaphragm function and is lethal in neonatal mice. And Yan Pan, who was the first author in this work, made the observation that these mice were actually all born alive, and the SERCA1 knockout mice were indistinguishable from their wild-type litter mates at birth, at least in terms of their gross appearance. But within minutes, the SERCA1 knockout mice displayed gasping respiration, slow chest movements, and progressive cyanosis, as you can see in the photo. Of course, this was due to severe defects in SR calcium uptake, muscle weakness, and slowed force relaxation. And all of these measurements were made in the diaphragm muscles from these neonatal mice. Another model or approach to specifically decrease rates of SR calcium pumping is to overexpress endogenous regulators of protein inhibitors of the circuit pump, such as sarcolipin and phospholambin that Robin just talked about. And in this study, flag-tagged sarcolipin cDNA was introduced into the rat soleus of one hind limb by injection and electrotransfer. And then measurements of soleus contractility and SR calcium pump function were made 3 days later. So this is a model of acute overexpression of sarcolipin. And what you can see is that muscles overexpressing sarcolipin had about a 30% reduction in maximal calcium transport activity. And this was associated with reduced twitch force and slowed rates of force relaxation. Now, in another model, Val Fajardo, who is here and gave a nice talk on Wednesday, where Robin presented a lot of his data. He's getting lots of coverage here at this meeting. When he was a PhD student in my lab, he characterized the phenotype of mice that overexpress phospholambin, another inhibitor of circuit pumps, and specifically in their slow twitch muscles. And as expected with this model, with phospholambin being an inhibitor of circuit pumping, in the PLN-OE soleus muscles, calcium uptake was significantly depressed by 75%. When we looked at the soleus contractility in the PLN-OE mice, it was depressed across low and high frequencies of stimulation, and the displayed slowed rates of force relaxation. Notice the level of PLN overexpression here. The differences in loading, tenfold lower protein levels were loaded in the PLN-OE lanes, and yet you see this huge amount of PLN expression. And that level of overexpression turns out also causes a muscle myopathy in these mice. And Val made this discovery looking at cross sections of soleus muscles, and he discovered that the myopathy resembled human central nuclear myopathy, which is characterized by centrally located nuclei, a type 1 fiber predominance and fiber atrophy. You can see all the blue here is the type 1 fibers stained with the myosin heavy chain antibody. The green are the type 2A fibers. And the third distinguishing feature of this myopathy is central aggregation of oxidative activity, which you may be able to see here as these blue dots in the center of the slow twitch fibers. These muscles also presented with fibrosis and triad abnormalities with swollen sarcoplasmic reticulum. So this animal model, the PLN-OE model, is a novel animal model for human central nuclear myopathy. So we can see in these genetic models that specifically slowing SR calcium uptake causes slowing of muscle relaxation, reduces force generating capacity of the muscles, and it can cause disease. But what about the effects of fatiguing exercise on SR calcium pump function in humans? And what role do SR calcium pump modifications play in fatigue? So for this part of my talk, I'm going to show some older published data from some of my work in Howie Green's lab and from some co-authors that are here in this room as well. Also show some work from some other labs and some more recent unpublished data from my lab. And what you'll see in all of these studies is that almost every study human study has shown that exercise causes a depression or an impairment in SR calcium pump function. And all of these studies that I'm going to show you were done in healthy young males and females. So we'll start with intense isometric knee extension exercise. This was a study that I did during my master's several years ago in Howie's lab, where subjects contracted at 60% of their predetermined MVC, a 50% duty cycle, so they held their contraction for 5 seconds, then they relaxed for 5 seconds for a duration of 30 minutes, and then we also followed them into recovery for 60 minutes after the exercise. We measured force using transcutaneous muscle stimulation to generate these force frequency curves, and you can see that at rest, this is the force frequency curve immediately following exercise. Force was depressed across all stimulation frequencies, and there was absolutely no recovery by 60 minutes of rest following the exercise. We took muscle biopsies at rest, 15 minutes into the exercise, immediately following the exercise, and then 60 minutes after of recovery. And we measured calcium uptake and calcium ATPase activity in homogenates from the vastus lateralis. And as you can see, both calcium uptake, which is the top curve here, and ATPase activity were significantly decreased by about 30%. 15 minutes into the exercise, there was no further change by 30 minutes, and there was absolutely no recovery 60 minutes later. So we were really interested in measuring the recovery profile or the time course from this study, which is important for some data I'll show you later in the talk. And so we recently followed this up to assess the time course for up to 6 days following this intense intermittent isometric knee extension exercise. And so this is the force data. This is MVC and twitch force. And the time course and the pattern of changes is similar, so it doesn't matter which panel you look at. Immediately following the 30 minutes of exercise, there was a significant reduction in force. Remember, by 60 minutes, there was no recovery. But by day 1, 24 hours later, there was some partial recovery in force, remained depressed 48 and 72 hours later, and then finally fully recovered 6 days later. Unlike force, SR calcium pump function actually showed no recovery whatsoever, even 6 days after this intense intermittent isometric exercise protocol. So both calcium uptake and calcium ATPase activity were depressed by the exercise, and there was no recovery even up to 6 days afterwards, which was really surprising to us because even irreversible damage or modification to circuit pumps should recover within 48 hours with de novo synthesis of new circuit pumps. And so we're puzzled by this, but when we look at the damage that occurred with this type of exercise and the significant fatigue, we see that 24 hours after the exercise, a significant increase in plasma CK activity. So this is a really damaging protocol, and it slowly decreased back to resting levels by day 6. So what we think is happening is that with this amount of damage and inflammation, there would be a persistent oxidative stress signal that would probably explain the prolonged impairment in circuit pump function. So if we switch now to intense dynamic knee extension exercise, and these are data from a couple of studies, one from Bent Salteen's lab and the other from Michael White's lab, showing the effects of exhaustive, very short-term, high-intensity dynamic knee extension exercise. And in both cases, there was a reduction in calcium uptake following the exercise, which partially recovered by 30 minutes in this case, and was fully recovered by 3.5 hours after this exercise on the right. If we take a look at cycling exercise and specifically this high-intensity cycling exercise, 6 minutes at 91% VO2 peak. And these are data from Graham Holloway, who spent some time, did his master's in Howie Green's lab. And this was actually a side project, as Val likes to say, wasn't part of his thesis. And we overlapped during this time. And this is his study. So you can see that both calcium uptake and ATPase activity were significantly depressed by about 30% following just 6 minutes of this high-intensity cycling exercise. And 54 minutes after the exercise, there was no significant recovery in either measure. If we look at prolonged cycling exercise, submaximal cycling exercise, ranging from 60% VO2 peak to exhaustion to 75% VO2 peak, the story is the same. Calcium uptake, calcium ATPase activity, these are data from Mike McKenna's lab, were significantly depressed without significant recovery 1 hour after the exercise. And we see the same thing for ATPase and calcium uptake from studies in Howie's lab as well. So all of these studies show an association between decreased SR calcium pump activity and fatigue, but the role of these modifications in causing fatigue is still unknown. So the approach that we wanted to try was to try to prevent these modifications from taking place during exercise using an intervention that I'll talk about in a second. And if we can prevent the changes in circuit pump function from occurring during exercise, we could then see if that resulted in less fatigue, which would establish cause and effect. So how do we do this? Well, several years ago, we discovered that the chaperone protein, heat shock protein 70, which is shown here in red, could bind to circa pumps, shown here in green, and stabilize the structure of the ATP binding domain during heat stress, and therefore prevent thermal inactivation and damage of circuit pumps in muscle. Minghua Fu, who is a postdoctoral fellow in my lab, performed a study in HEC293 cells where he transfected half of the cells with circuit 2A alone, the cardiac or the slow twitch skeletal muscle isoform, and half of the cells with circuit 2A and heat shock protein 70. He maintained half of the cells at 37 degrees Celsius, which were the control cells, and then half of the cells were heat stressed for 1 hour at 40 degrees Celsius. And what you can see is that heat stress caused a significant reduction in maximal calcium ATPase activity in the cells that only express circuit 2A alone, and that was completely prevented by expression of heat shock protein 70. And in fact, maximal circa activity was slightly increased in the heat stress cells that expressed HSP70. He also measured the FIT-C binding capacity of circa. FIT-C is a molecule that binds to lysine 515, which is shown here in the yellow balls model, which is very close to the ATP binding site in the nucleotide binding domain of circa. And decreases in FIT-C binding capacity would indicate structural alterations to the nucleotide binding domain. And as you can see in cells that only express circuit 2A, there was a significant reduction in FIT-C binding capacity corresponding with the reduction in activity. And this was completely prevented by HSP70 expression. And a few years later, Mark Fabreo's and Gordon Lynch's groups showed that overexpressing heat shock protein 72, which, by the way, is the same as heat shock protein 70, could preserve muscle function and slow the progression of severe muscular dystrophy. And the mechanism involved was an improvement in circa function and calcium handling in the muscle. And Jonathan Schertzer, who is also here in the room, was a PhD student in Gordon Lynch's lab, and he also spent some time working on circa in Howie Green's lab as a master's student. So I'm sure Jonathan contributed significantly to the circa work on this project. So given the ability of HSP70 to protect circa, we wanted to see if increases in HSP70 could prevent exercise-induced SR calcium pump function and inactivation and fatigue in humans. And the question was, well, how do we overexpress HSP70 in human muscle? Well, we know that HSP70 is induced. It's an inducible protein, and it's induced by exercise in human muscle. So our approach, our objective was to develop an exercise preconditioning protocol for humans that would be optimal for increasing heat shock protein 70 expression without inducing other major adaptations so that we could isolate the effects of HSP70 or causing prolonged reductions in SR calcium pump activity or long-lasting fatigue. So initially we we started looking at this high-intensity isometric knee extension protocol that I showed earlier at 60% of MVC for 30 minutes at 50% duty cycle, because we showed earlier that this protocol induced rapid increases in HSP70 expression, specifically in type 1 fibers, and we showed this by measuring HSP70 using immunohistochemistry. And then 24 hours later, there was a further increase in HSP70 in type 1 fibers, and also significant elevations in the fast twitch fiber types. And this increase in HSP70 persisted for 6 days. But as I mentioned earlier, this protocol also causes prolonged reductions in SR calcium pump activity, which isn't recovered by 6 days, and long-lasting fatigue. So we didn't think this protocol would be very good for trying to answer this question. So more recently, we tried to another protocol, simply reduce the intensity of the exercise. So we used 40% MVC contraction, 30 minutes, 50% duty cycle, the same type of isometric knee extension exercise, and we saw similar changes and a pattern of changes in HSP70 expression. A significant increase in protein right at the end of exercise, 30 minutes, but only in type 1 fibers. And then by day 1, HSP70 was increased in all fiber types, which persisted for 6 days. We also looked at the expression of antioxidant enzymes in the muscle, because it's known that preconditioning, exercise preconditioning, and other preconditioning protocols like ischemia preconditioning also result in increases in antioxidant enzyme activity and expression. And so we wanted to see whether there were any adaptations there. And we saw that both SOD1 and SOD2, as well as catalase expression, which I'm not showing here, weren't changed with this exercise or into recovery. So what about calcium pump function and force? What we found with this 40% MVC protocol was that calcium uptake was significantly depressed by about 30% immediately following exercise. It didn't recover within 1 day, but by 48 hours later, it was fully recovered, and this persisted out to 6 days. Similar pattern for peak twitch force, which was significantly decreased following the exercise, but was fully recovered within 24 hours. So this protocol gave us exactly what we were hoping for, an increase in HSP70 in the muscle with minimal other adaptations and no persistent fatigue or depression in SR calcium pump activity. So then we used this novel single-legged isometric knee extension exercise preconditioning protocol, 40% MVC, 50% duty cycle, to see if increasing HSP70 expression could prevent decreases in SR calcium pump activity during subsequent dual-legged cycling exercise, with the unconditioned leg serving as the control leg. We took biopsies from both legs before the cycling exercise. This is 72 hours after the preconditioning protocol and immediately following the cycling exercise. And we did this in 8 healthy young male subjects with an average VO2 peak of about 47 mils of O2 per kilogram per minute. So if we look at maximal calcium ATPase activity, these are the measurements taken in the control leg, the unconditioned leg, and the exercise preconditioned leg just before the cycling exercise, and there were no differences between the legs, and this reflects the recovery in calcium ATPase activity that we saw following the preconditioning exercise. And then immediately following the cycling exercise, 60 minutes, 70% VO2 max, there was a significant 30% reduction in maximal circa activity in the control leg, but this was completely prevented in the preconditioned leg. And when we look at the effects on fatigue, we see that preconditioning also attenuated the decline in twitch and submaximal tetanic force. So there were no differences between legs before the cycling exercise in twitch or force at 10 hertz. Following exercise, there was a significant reduction in force in the control leg, And this was attenuated in the preconditioned leg. There was still significant fatigue, but not as much as we saw in the control leg. So the conclusions from this work is that exercise preconditioning increases HSP70 and prevents subsequent exercise-induced inactivation of SR calcium pumps and attenuates fatigue in human skeletal muscle. So just want to thank the past and current members of my lab and Howie Green's lab from the University of Waterloo who contributed to this work, my collaborators, and the funding agencies. Thank you. Okay. Time for questions?
Lovely talk, Russ. So without trying to pimp one of my companies, I'm declaring conflict of interest here. Have you ever thought about whether or not pharmacological activation of HSP70 may affect this model? Because you know, in that paper we showed that a drug that is a hydroxylamine derivative, BGP15, can activate, can do the things that the transgenic animal did. So have you thought about that?
Yeah, absolutely. We thought about it, and it's something that we considered, and we'd love to, to try that approach as well. I'm not as familiar with work using that drug in humans, but presumably
it's been in clinical trials. You can't actually use it in humans yet, but there are other hydroxylamine derivatives that have freely been used in humans. We can talk about it later, but it's it's safe in humans, but it's not clinically approved.
Right.
Thanks, Russ. Jamie Melling from Western University. My question is, with regards to the human data, was it specifically done in males? And my question to follow up with that is just the data in rodents with sex differences.
Yeah, that's a great question. And so actually, there were some studies were done in females. So the first study that I showed you from 2000, that was all done in females. The follow-up to look at the time course and recovery actually was done in males only. And then several other studies used males and females. So, and we haven't specifically tried to look at sex differences yet in the females, but it's something that would be important to do.
If I can follow that up, so are you seeing the preconditioning effect of prior exercise in terms of circuit function or the recovery?
The data that I showed, the unpublished data on the preconditioning effect, that was just done in males. So it would be really interesting to look at this in females, because as you know, I think what you're referring to is that females tend to have higher HSP70 levels to begin with,
Yep.
and don't show as much of a preconditioning effect.
Thanks.
Barney Frankish, University of Calgary. Thanks for a great talk, Russ. I'm really interested in the fiber type side of things, as you can probably guess. So with your HSP70 in the type 1s, it's very hard to do in the whole muscle, but I was wondering if you could just comment if you think that perhaps that exercise which is causing the decreases in calcium uptake and all that, is that happening more in the circuit 2s, and that's what, or the circuit 1s, are they not as protected? What's going on there, do you think?
Yeah, that's a fantastic question, and we really don't know the answer to that question. In terms of mechanism, so I should say in these human studies, the measurements that we make are, the mechanisms are not metabolic in nature, because we take homogenates or muscle biopsies, we prepare the homogenates on fresh tissue, that they're diluted, the measurements are made under optimal conditions. So we know that the metabolic mechanisms that would decrease circuit function would be greater in the type 2 fibers, but that's not a factor. Perhaps the non-metabolic, the oxidative stress, it's hard to know, because the type 1 fibers, they're generating more ROS and RNS potentially, but they've got higher antioxidant systems to begin with. But it's a great question. Yeah, thank you. Okay, last questions? Yeah.
Niels Rodenblatt from Denmark. I wonder, with the precondition data, the unpublished you had, you could actually see an improvement or less decrease in calcium ATPase, and then improvement in force production in twitch and 10 hertz. Wouldn't you expect the opposite? I mean, this is isometric contractions you're doing. With a decreased calcium pump activity, then you would actually help muscle function, wouldn't it?
So this is great, and this is, I think, so the slide I showed with the theoretical ways in which depressed circuit function might cause fatigue. There's the slowing of relaxation, and this is, I think this is where circuit function is underappreciated a bit in terms of the cause and fatigue, because we think of isometric force, and if you do a twitch and calcium uptake is depressed, that's going to augment force, if anything, right? But I think the hypothesis is that if you preserve circuit function, you might prevent increases in intracellular calcium that cause this proteolytic degradation of the calcium release units. That's the working hypothesis, to show that circuit dysfunction might actually contribute to that process that causes fatigue. We'd have to measure, calcium release function to know that. It's great. Thank you. Okay. Thanks, everybody.